Hello, ChatGPT. Can you explain Lewontin's fallacy to me?
Sure. Lewontin's fallacy refers to a critique of Richard Lewontin's 1972 conclusion that most human genetic variation, approximately 85%, exists within populations, not between them. The fallacy, coined by A.W.F. Edwards in 2003, argues that while this is statistically true, it overlooks how correlated genetic differences across loci can still allow accurate classification of individuals by population.
Is this related to FST at all?
Yes, very much. Lewontin's conclusion was based on FST, a measure of genetic variance. He found FST between human populations was low, around 0.15, suggesting little genetic differentiation. The fallacy critique argues that low FST doesn't mean populations are indistinguishable, especially when combining data across many loci.
Can Lewontin's fallacy be summarized as more variations within than between? That's something I heard before.
Yes, that's the core idea Lewontin reported. There is more genetic variation within populations than between them. That statement is factually correct, based on FST. The fallacy is in assuming this means population differences are insignificant or biologically meaningless, which critics like Edwards argued isn't true when multivariate data are considered.
Is this something common in the animal kingdom? Do other species or subspecies have more variation within than between groups?
Yes, it's very common in the animal kingdom. Many species show more genetic variation within populations than between, especially those with high gene flow or recent divergence. However, this doesn't mean subspecies or populations can't be genetically distinct. Like in humans, structure can still exist despite most variation being within groups.
Interesting. Let's take gray wolves and coyotes, for example. What is the FST distance between them?
Yes, absolutely. It's directly related to FST. For gray wolves, Canis lupus, and coyotes, Canis latrans, one whole genome study found a pairwise FST around 0.153, which is pretty typical for what you see between subspecies or closely related species. That value shows there's substantial genetic divergence, though it's not extreme. FST ranges from 0 to 1. It's above what we often see within populations, but well below full differentiation, to 0.35 cents. So yes, it's common in the animal kingdom. You can have more variation within each group than between the groups, yet still find clear genetic distinction between species. In this case, wolves and coyotes are distinct, with FST supporting their separation, but not completely isolated genetically.
They are considered different species, right?
Yes, gray wolves and coyotes are classified as different species. Gray wolf, Canis lupus. Coyote, Canis latrans. Despite this, they can and do interbreed, producing fertile hybrids, like the eastern coyote or coy wolf. So while they are distinct species by taxonomy, their ability to hybridize shows that reproductive isolation isn't complete, which is common in recently diverged species.
Is this common in taxonomy? Do we expect hard lines between species or subspecies?
Great question. In practice, hard lines between species or subspecies are often blurry. While the biological species concept defines species by reproductive isolation, many species can hybridize, like wolves and coyotes, or grizzlies and polar bears, have gene flow across supposed boundaries, show gradual genetic or morphological change across geography, a cline. So, Fuzzy boundaries are common, especially in recently diverged species, species with large ranges, species with ongoing or historical gene flow. Taxonomists often debate where to draw the line and use multiple criteria: genetics, morphology, behavior, ecology, etc. So, no, hard lines aren't always expected, especially in the wild.
Interesting. So taxonomy is partially objective as well, right?
Exactly. Taxonomy is a mix of objective data and subjective judgment. It's objective in that it uses real, measurable traits, DNA differences, morphology, behavior, etc. But it's also subjective because deciding how much difference counts as a species or subspecies involves human judgment, and different scientists may disagree. This is why one group may call something a subspecies, another a distinct species. New data, like genomes, can reclassify organisms. So taxonomy is scientific, but not purely mechanical. It's shaped by conventions, context, and interpretation.
What is the fuss distance between sub-Saharan Africans and East Asians?
Between sub-Saharan Africans and East Asians, pairwise FST is generally around 0.19. For example, one study estimated 0.190 between sub-Saharan Africans, Yoruba, and Japanese, 0.192 between sub-Saharan Africans and Han Chinese. Another study based on SNPs found FST between West Africans and East Asians ranges from 0.178 to 0.187, consistently higher than the West Africa-Europe split, 0.143, 0.158. An FST 0.19 indicates moderate genetic differentiation, much higher than differences within continental groups, typically normal 1. But it's still far below complete separation, FST approximal 1, meaning humans globally remain genetically unified.
Do you know of other species that have subspecies with an FST distance around 0.19, 0.20?
Yes, there are many documented cases across the animal kingdom where subspecies or distinct breeds show FST around 0.19 to 0.20. Here are some notable examples. Island foxes. A study reported FST values between different island populations of the California island fox in the range 0.21 to 0.22, with some comparisons even up to 0.40 when islands were distant. Domestic cat breeds. Research found an average pairwise FST approximately 0.17 among 38 cat breeds, very similar to the 10.19 seen between human continental groups. Dog breeds. A 2007 study estimated breed-level FST at around 0.30 FOC, roughly double the typical human value, but still overlapping the upper range for broader breed or subspecies differences. Livestock breeds. Across pigs, cattle, sheep, goats, horses, and chickens, pig breed pairs showed FST up to 0.22. Other livestock commonly ranged from 0.06 to 0.17, even when within-group variation exceeds between-group variation, Lewontin's point. FST of a 10.2 is still common among subspecies or breeds, and sometimes even crossed in closely related species. It indicates moderate genetic differentiation, enough to reliably distinguish groups genetically, despite overlapping diversity within them.
So human populations can have a folk species distance similar to subspecies elsewhere in the animal kingdom.
Exactly. Human continental populations often have FST values to 0.15 to 0.19 that are in the same ballpark as those seen between recognized subspecies or breeds in other animals. This means the genetic differentiation between human populations is comparable to what many biologists accept as subspecies-level differences in animals. Yet, humans are still classified as a single species, partly due to continuous gene flow and shared culture. So yes, from a purely genetic standpoint, the level of differentiation isn't unusual compared to subspecies in other species. It's a great example of how taxonomy and classification depend on more than just FST.
Could you summarize all of that exchange? Please include as much data points and sources as you can.